about this, I just want to mention that although I'm a yeast geneticist, I'm not really so much interested in yeast itself, sorry to say. I'm more interested in using yeast to answer questions and study questions that are universal. And what can be more universal than DNA, right? So as shown here in the title page right here, you see the famous double helix. There are two strands of DNA that are interacting with each other, forming a right-handed double helix. And this double helical structure is only possible through the interaction of the two strands through the base pairing of complementary bases like A and T and G and C. However, there's always an however, right? These DNA can form structures that are beyond these double helical structures. So double helical structures for historical reasons are called B-DNA. And there are many of non-canonical, non-B-DNA secondary structures that DNA can assume. And here are a few examples shown. And one of these are hairpin structures. Within this single strand, blue strand, there are base pairings. So if you have sequences are reverse and complement, you can form and extrude as a hairpin. If you have a strand of sequence that contains many, many purines, they can actually have three of the DNA strands interact with each other to form something called triplex DNA or HDNA. For today's talk, I'm going to focus on another structure, sometimes referred to as a four-stranded DNA, because it looks like there are four different DNA strands involved, but this is actually made up of a single strand that are interacting in a very weird way. And this very weird way is actually four guanine bases interacting with each other, forming a ring or a square-like structure, depending on how you see it. And this is called G-tetrad. And when a strand of DNA contains multiple runs of guanines, like sequence shown here, then multiple of these G-tetrad, each one of these planar form is G-tetrad, can form, and these multiple tetrads can stack on top of each other to result in a very stable structure called G4-DNA or G-quadruplex DNA. And obviously, this is not, just like all of us who are very familiar with double-strand DNA structures, cellular processes that involve DNA, like transcription, replication, and repair, are supposed to be more familiar with double helical structures and not the G4-DNA. So when these G4-DNAs then interfere with these processes, result in something called genome instability. So when I say genome instability, what do I really mean? And they include a whole lot of different things. And obvious thing that people recognize are mutations, that is single nucleotide variations. There can also be a very large change, like number of the chromosomes can change down to monosomes and trisomes. And also you can have small parts of the chromosome change copy numbers through deletions and amplifications. There are also a very unique type of genome instability called translocations. When DNA is broken, either through double-strand break or single-strand break, different parts of the different chromosomes, so here are two different chromosomes, can actually try to join ends in a way it results in this kind of mishmash mixture of chromosomes. Now, aberrant chromosomes are created through balanced translocation in this case. So translocations in particular, but mutations and other types of genome instability, like I mentioned, all occur in abundance to lead to cancer. And also cancer genomes, when you sequence them, contain many evidences of these genome instability events. And what people have been finding through bioinformatics study of cancer genomes recently is that cancer genomes contain lots of these mutations and recombinations and translocations everywhere in the genome, but specifically, they are very highly enriched at places where they contain very specific sequences that has a potential to form G4-DNA. So sequences that contain G runs are more prone to mutations and recombinations and translocations. So focusing on translocations, so here are some of the genes where translocations in blood cancers have been shown to occur in recurrent way, so that is multiple in many different individuals. These include IgS mu sequence, BCL2, c-MYC gene, TCF3 gene. What all these sequences have in common is that they have a lot of translocations occur, obviously, like I said, but also that they contain G4 motifs. But these bioinformatics study that I talked about so far only showed you the evidence of something happening after the fact. It does not tell you anything about the mechanism of how these things occur, and does not tell you about what other factors, like what other proteins might be involved in having these recombinations and translocations occur at these locations. So what we want to do is we want to use the yeast to answer the mechanistic details about these events occurring at G4. So what we did is we took one of these sequences, S-mu sequence, I'm going to refer to a lot, that contains lots of guanine runs as shown here that can form G4 DNA, and put it into yeast genome, specifically on chromosome 3 of yeast genome within the context of a lyse2 gene that is transcribed from a PTEP promoter that's regulated. So this red box, for example, is representing the G4 forming S-mu sequence. If break occurs at this red box, then it can be repaired through recombination with another piece of DNA somewhere else, but that recombination results in a phenotypic change from lyse minus to lyse plus that we can score, so we can actually measure and answer quantitatively the recombination rate occurring at this gene. And that will be a representative of how often breaks occur at this location. So another feature of this reporter assay is that this red box was integrated into the genome in two different orientations respect to the transcription of the lyse2 gene. So transcription occurs, here is the polymerase complex moving this way, and here's a red line which is representing the RNA. In G-top orientation, I'm going to talk about G-top, G-bottom a lot. G-top orientation, guanines, or the G's on the top strand, so it's left as a single strand DNA during transcription temporarily. Whereas, in the G-bottom configuration, it's on the bottom strand that's being transcribed, and it will be base paired with nascent RNA and is not left alone. So the guanine bases are now free to interact with each other to form the G tetrad that can lead to formation of G4. So we would, if the G4 DNA leads to recombination, then we would expect the G-top will be more prone to recombination, especially if it's transcribed. And that is true, it's shown in the recombination rate we measured. When the transcription is turned off, the recombination rate at G-top and G-bottom are the same and low. When the transcription is turned on, there's a general effect of transcription and recombination rate is elevated for both G-top and G-bottom, but they're significantly higher for G-top. This indicates that G4 DNA forming more prone to being formed in G-top orientation during transcription is leading to higher recombination, higher breaks in DNA, higher recombination. So now we're using this system to study what factors are involved in affecting this genomic stability occurring at G4. So here is a very simple example of TOP1. TOP1 is a topoisomerase 1 that relieves excess helical torsion accumulated by unwinding or overwinding of DNA. When TOP1 is deleted, compared to wild type, there is a change in recombination rate for G-top orientation only, but there is no change in recombination for G-bottom. This says that it is specific for G4 formation, and there's somehow increasing, deletion of TOP1 is increasing the formation of G4, leading to higher recombination at this reporter. So this is basically the principle we're going to be using for the result that we're going to talk about today, to show or to identify and characterize factors that are affecting G4 DNA instability. So G4 blocks TOP1 blocks G4 DNA formation and reduces and suppresses G4 DNA associated recombination by removing negative torsions that are introduced during transcription. And we are going to talk about examples of two different types of G4 associated protein. So these are going to be all G4 binding proteins that either, first of all, resolve G4 structures, thereby reducing and suppressing G4-induced genomic stability. When this factor is gone, obviously, that the recombination rate at G4 will be higher. But the other types of proteins will be G4 binding proteins. And by binding to G4, it will be stabilizing the G4 structure. It will enhance G4-induced genomic stability. When this factor is gone, recombination rate will be lower. So I'm going to start talking about the example of the first class, which is sub1. It's just a very small transcriptional cofactor, and it has a highly conserved homolog, human homolog called PC4. It was identified in 2015 as one of the protein, actually the only protein that was shown to bind to G4 DNA forming oligos in a pull-down mass spec assay. So this is one of the most highly G4 specific binding protein in the whole yeast cell extract. When you delete this protein, like I talked about, the principle of it, when you delete the sub1, you have an increase in recombination rate. And this recombination rate increase is only specific for G-top and not G-bottom. So it's a specific for G4. And I want to say one thing that most of the assays we're going to talk about, we're using top-1 deletion background so to accentuate the difference between G-top and G-bottom. So I talked about the human homolog that's highly conserved. And when you have a sub-1 deletion background, you can actually supplement sub-1 expression or human PC4 expression to lower the recombination rate back down. And when you do chromatin IP, you can see that sub-1 is enriched at G4 forming sequences, but only in the G-top orientation and not in G-bottom orientation. And they're only enriched during high transcription and not when the transcription is turned off. So this says that sub-1 is binding to G4 DNA, but not the G-run forming sequences, but G4 structure itself, because it is only binding to G-top. G-bottom, again, because it has a G strand and C strand, does have all the same sequences as G-top. So I'm going to very briefly talk about what we figured out, how this very small protein is doing this, suppressing G4 associated instability. And through a collaborator, Dr. Rainey, we found out that sub-1 is interacting with another protein called PIF1. So this is a co-IP assay using yeast whole sac extract. And this is an assay used showing interaction between the two proteins using purified sub-1 and purified PIF1 in both cases. And also genetically, we saw that deletion of sub-1 and deletion of PIF1 each results in increase in recombination rate at G-top. But this rate is actually the same as a double deletion of sub-1 and PIF1. This is a very classical epistatic relationship, which shows that PIF1 and sub-1 actually function together. And by showing that they interacted with each other physically as well, we're showing that they must be working together as a protein complex. So what could this be doing? And this obviously need to tell you that PIF1 is actually a DNA helicase that unwinds G4 DNA structure. So what we think is that sub-1 can bind to both the G4 DNA and PIF1. And by this characteristic, it helps recruit PIF1 to G4 DNA, where PIF1 can now function to unwind G4 DNA to suppress genomic stability associated with this. So now for an example of the second class where exactly the opposite happens. And it's a protein called NSR1 in yeast, and it's a homolog of human protein called nucleolin, which is a very abundant protein in nucleolus, which is a structure shown here within a nucleus. And it has it's a multifunctional protein. And it has been, its human homolog and actually mouse homolog were shown very early on to be a very specific G4 binding protein. So it binds to many different G4 sequences, including the IgS-mu sequence, C-mix sequence, and the GGGGCC repeat sequence that are associated with ALS. So again, like I talked about before, if it's a protein that binds to G4 and by binding increases its stability, then when we delete NSR1, we would see reduction in the recombination rate occurring at G4. And this is what we saw. Again, it's only specific for G-top and not G-bottom. And again, we just like sub-1, we tested where it can bind to G4 DNA. we used 3 different oligos, one that forms G4 and 2 where we disrupted guanine run, so it doesn't no longer form G4. And what we saw was that it really only specified very high affinity binding with the SMUG G4 forming oligos, and not as much to the other 2. And when we deleted this very C-terminal end sequence, RGG domain, so to create a NSI1 delta RGG truncation, it no longer binds to SMUG very efficiently. And we did this because it was known for the mouse protein that this C-terminal end domain is necessary for very high affinity binding to G4. And this was also true for yeast. And we again did the chromatin IP, just like the sub1, and we showed that full-length NSR1 is enriched at G4, but not the truncation. And again, at the G bottom construct, it's not enriched at all. So it is a G4 DNA specific binding. So what happens after NSR1 binds to G4? One idea we had was that this can bind very specifically, form a struct complex that is very stable, and becomes a block to replication. So in order to test this, Dr. Shivani Singh in our lab has developed a method that's a modified ddPCR. And ddPCR is a form of quantitative real-time PCR. And it's a very sensitive method, so it can distinguish between very small differences in copy number of the target gene. So what we did was we arrested cells in G1S, and then release them into the S phase in a synchronized way. So all the cells begin replication in a synchronized way. Take samples at 10-minute intervals. DNA is isolated and sonicated, and copy number of a target location is determined by ddPCR. So all our cells are haploid yeast, so they'll start with 1 copy of every single location in the genome. As they're released into S phase, as they progress through the S phase, they'll eventually start replication, and different location within the genome will actually replicate at different time. But eventually they'll all reach copy number 2, and then they'll divide. So if you look at this as single location, you'll start with a copy number 1. Eventually, for this location, it starts replication about 15 minutes, finishes about 70 minutes, which is copy number 2 at 70 minutes. So if you do this for 3 different locations around our G4 sequence, so R306 is where the replication starts. Ours is a replication starting sequence, and another location upstream, and then another location that replication has to go through the G4, and then it reaches this location as T50. At R306, and also at KNMX, you can see that the replication progresses at the same kinetics, whether you have the full-length NSR1 or not. That is, R306, whether you have full-length NSR1 or not, it's reaching copy number 2 at around 15 minutes after release. However, at the location SC50, which is location replication fork has to go through G4 to reach, you see that there's a big difference in replication kinetic. If you have a full-length NSR1, there's about 20-minute delay in the time it requires to reach the copy number 2. So that means having full-length NSR1 bound to G4, which we verify through the chromatin IP, results in delay in replication, problem for the replication. That's very significant, so it delays replication by 20 minutes. But what happens if you have the same sequence, same transcription, but in the G bottom orientation? Now again, everything's the same except G4 DNA cannot form. And you can see at ST50, there is no longer that significant 20-minute delay in replication kinetics, whether you have full-length NSR1 or not. So it seems that NSR1 is actually forming a very high affinity complex with G4 and obstructing replication, leading to genomic stability. So final example today that we're going to talk about is actually back to that very first protein we talked about, which is topoisomerase 1. Topoisomerase 1, first of all, binds to DNA, double-strand DNA. And then second, it cleaves one of the strand. And the third, actually ligates it back together. And the resulting product is actually same piece of double-strand DNA, but has less of the helical torsions it had at the beginning. So remember the 3 functions: binding, cleaving, and ligating. So it's a very long protein and very highly conserved protein, but it's very important catalytic residues are present in the C terminus, at the very end of C terminus. So it's because of this role of relieving helical torsion that I introduced when the DNA strands are separated. It is an essential protein in human, and it is a target of anti-cancer drugs of CPT family. And cancer patients and cancer cells that are treated with this drug develop resistance to this drug by mutating its target protein, which is TOP1. So we wanted to see what would be the effect of expressing some of these mutants in E cells and looking at the recombination occurring at G4. So we're expressing 3 different mutants. It's going to be kind of wordy for me to like say all these names, so I hope you can follow me. But Y727F is a catalytic mutant that has the very important catalytic tyrosine mutated. It's found in the screen for a catalytic dead mutant. But the other 2 mutant, Y740 stop and S733E, are mutants that are analogous to the human mutants that are found in cancer cells that are treated with drug and develop resistance. So these 2 are cancer resistant drugs, like cancer and CPT resistant mutations. All 3 are actually CPT resistant mutations. But there are differences. The ones in red actually can still bind DNA, just cannot cleave DNA. Whereas the one in blue, S733E, cannot even bind DNA. So whether it can cleave DNA or not is irrelevant. When we express this in top-1 deletion strain, first of all, a review of top-1 deletion, complete null of top-1 increases recombination rate only in GTOP. And now it's about 107. For when we express S733E mutant in place of wild type top-1, mutation is only elevated for GTOP, but up to about 72.3. With error bars, this is statistically the same rate as complete null of top-1, which makes sense. You have a dysfunctional mutant expressed in place of wild type. Obviously, it's going to act like top-1 deletion. So obviously, that's not where the surprise comes. Surprise comes when you express these 2 red mutants that can still bind DNA, Y727F and Y740 stop. The recombination rate is elevated for GTOP, but way, way beyond what is the rate for the top-1, complete deletion of top-1. So obviously, these 2 mutants specifically has gained some kind of function that is leading to aggravated recombination rate at G4 sequences. So just like all the other protein, first thing we do is whether they can actually bind to G4. So this is the same oligo pull down assay. SMUG is a G4 forming, M1 is not. And you can see that wild type top-1 binds to G4. Y727F, Y740 stop binds to G4 and not the control. But S733 does not bind to G4 oligos. So binding to G4 is what's distinguishing these 2 mutants that increase recombination rate compared to this one, where it does not bind to DNA. And these 2 mutants bind to G4 in a potassium-dependent way. When you replace the potassium with a lithium cation, it no longer binds to G4. And lithium ion was shown before to disrupt the interaction between or among guanine bases to form that G tetra that's the basis of G4 DNA. So this shows that these 2 mutants are binding to this SMUG oligo, not in a sequence-specific way, but structure-specific way that G4 DNA structure has to form in order for these to bind. So again, just like a very similar way to nucleolin, we hypothesized that top-1 mutants binding to G4 DNA, forming a very stable complex leading to replication block. So for protein that is binding very specifically or even covalently crossing to DNA that are stuck on DNA, there is a pathway called protease-dependent repair that can actually recognize these proteins that are stuck on DNA and actually degrade the protein to get rid of it. And for this to happen, first of all, this protein has to be sumo-related, modified by adding sumo protein onto the protein. And Spartan, or WSS1, which WSS1 is the yeast name for Spartan, these are metalloproteases that recognize these sumo-related proteins on DNA and comes and degrades these proteins. And that way it relieves that replication block caused by these protein DNA crossings or protein DNA complexes. So we thought that if the topo mutants are forming these protein DNA complex that are stuck on DNA, then first of all, it will be sumoylated. And second, it will be a substrate of WSS1 in yeast. So for the first part. So Ellie expressed his tagged sumoprotein in yeast cells and pulled down all of his sumoylated proteins and looked for whether topo proteins and topo mutants are sumoylated. And you can see here, these mutants, including S733E mutant, are actually showing this higher molecular weight bands and some of these multiple bands, and they are actually sumoylated, either mono or poly-sumoylated forms of the protein. So these proteins can be sumoylated, and we don't know what the significance of S733E mutant being sumoylated because it actually does not bind to DNA. But moving forward through genetic work, or to the genetic work, if you delete WSS1, either in wild type, topo deletion, or cells expressing topo S733E mutant, there is no change in recombination. However, in cells expressing Y727F or Y740 mutants, there is a significant increase in recombination rate when you delete WSS1, which means that these proteins are actually are targets of WSS1 protease normally, that although WSS1 does not completely remove the effect of these mutants on G4 DNA, they partially can relieve some of the effects. And finally, I just want to mention one piece of data, and that is that in collaboration with John Taylor's lab at MD Anderson, we looked at about 35,000 cancer genome samples from Cosmic Database, and we identified 239 samples of these that has mutations in TOP1. What we wanted to see was having mutations in TOP1 does then increase the chance of having higher mutation rates, and especially at G4 forming sequences in the genome. And that was true. Not shown here is that mutations in TOP1 is correlated with generally higher mutation rate everywhere in the genome in the cancer samples, cancer genome samples. And here what we're showing is that when you have mutations in TOP1, and they're actually divided into different domains, but altogether having mutations in TOP1 means that there is higher percentage of mutations at G4 forming sequences compared to random samples. And this is especially true for the mutations in the C-terminal domain of TOP1. Like I pointed out earlier, the very important catalytic residues of TOP1 are present at the C-terminal end. And this has the highest percent of mutations in G4. So that means the cancers with mutations in TOP1 gene are correlated with higher mutation rates at G4 forming sequences in the genome. So I showed you only some of the data because this is going to be a short talk, but this basically says the topo mutants, the ones that still retains ability to bind to DNA, does bind to G4 formation, G4 DNA that are formed in the genome and form a very stable complex, blocking replication, leading to genomic stability and recombination. And obviously, we haven't shown the changes in replication kinetics just like we did for the NSR1, and we're still working on that. In addition to that, we're working to try to get more details about the mechanisms underlying how sub1 functions, NSR1 functions, or topo mutancy functions. And that's the current work. So I showed you the examples of two different classes of G4 binding proteins. One that can actually help reduce the recombination occurring at G4, one that actually increases recombination occurring at G4. So here are some of the lab members that worked on it. Three people I want to mention is that Chris Lopez has worked on sub1 story. Shivani Singh has worked on NSR1 story. Ollie Burrow has worked on topo mutation story. Ollie will be defending her work in 3 weeks. So if you're interested in more details about this, the topo mutant work, that you can come to her defense virtually, I think. And I'd like to thank all the collaborators and the funding source, and I'll take any questions. Are there questions in the chat?
Yes, we have one question in the chat so far. So Jalissa is asking, for the first mechanism for alleviating G4, the two proteins NSR1 and PIF1 were recruited. One acts as in helicase, is it acting to relieve the G4, or is the G quadruplex resolved first?
You mean sub1 in PIF1, but sub1
Okay.
is, it by sub does not have any function in unwinding G4. But PIF1 is known, and it's been very well characterized in vitro, to be a DNA helicase that can actually unwind G4 back to, you could say, single-strand DNA, so that double-strand DNA can be reestablished. So I'm not really sure what it means by relieve G4 or resolve G4. I think they're both the same, referring to the same thing. So yes, it is the PIF1 that's actually carrying out its catalytic function as a helicase to resolve G4. But it's sub1 that binds to G4 and help bring PIF1 to G4 DNA.
Okay, thank you. So I guess I was asking in the sense that you have this very stable structure with these G4s forming this quadruplex, and then they get stacked on each other. You were mentioning that the PIF1 acts on the top mostly. So I was wondering, because the G4 is so stable, was there something that helped to like break it apart a little bit before it gets unwound, or is it that PIF1 actually comes in and do most of that work?
PIF1 will by itself to do it. In vitro, people have shown that PIF1 by itself will very efficiently unwind all the G4 structures.
Okay, thank you.
Yes. Any other question? All right, great. Thank you.
Thank you so much, Dr.
I'll
Kim.
stop sharing.